Okay, welcome to our next episode of our phytochemistry. Now we move on to the next chapter of our Phytochemistry 2, looking at different evaluation of plants. Now, let's try to summarize first in just 5 minutes of time. So looking at all that we have learned from our summary last time, Phytochemistry 1, we have tackled about adulterated drugs which come up in your examination. And some of you may answer it or may have difficulty of answering some of the evaluation questions and analytical questions. Perhaps we say that adulterated products are those which are spoiled already, or maybe during roasting, or maybe application of heat in terms of storage, in terms of, for example, intentional addition of a certain product or admixture of a certain product. And of course, aside from that would be unintentional, which is depending upon, like for example, some factors. Number one would be in terms of storage condition, in terms of heat, in terms of the process that we undergo during extraction and during evaporation, during heating. No, these are the processes which are viable to certain heat or decomposition, or shall we say oxidation reduction, right? So there are different factors that affects in terms of drug adulteration. Now we move on to the different types, and this reflect in your examination whether it is direct or indirect, or shall we say unintentional. So the main difference of these two is that intentional, we have the purpose to reduce in terms of what? Cost, and adding up the profit. No? So meaning as a businessman, you just, instead of meat that you're going to put, or for example chicken, you try to put tofu, which is similar as chicken, right? That is adding up in terms of the profit, but lesser in terms of capital, or lesser in terms of cost. Okay? Because, of course, as a pharmacist, we look at always what is the cost of a certain product. Example of which is, the very costly one is what? The perfumes, essential oils, which is coming from a natural product. Once it is in a natural product, then it is very costly to extract certain minimal percentage of essential oil to a certain plant. So now, what are you going to do? To top up another mixture, or to replace or substitute another set of ingredients so that your product will have lesser cost but the same in terms of what? Appearance, color, maybe smell or aroma, no? Its particular texture, no? Morphological characteristic may be similar. Okay? So, but then in terms of quality would be lesser. Okay? So in that sense, as what our case have mentioned in the examination, it was brought into the pasamalam, right? And of course, if that be pasamalam, what will be? The quality. Okay? What is that quality are we looking at? Right? So if that will be, not to mention, if that will be in the herbal shop, we cannot categorize which one, right? What if in case, for example, adverse effect that will happen to the patient, we may say that the quality may be what? Causing this kind of adverse drug effects, because it may cause adulteration, or it may cause idiosyncrasy to a particular patient. Okay? So that one. So these are the main essence of our phytochemistry 1. So we're going to move on to our phytochemistry 2. Looking at, in our phytochemistry 2, this is a continuation of adulteration or adulterated products. Okay? So looking at our Phytochemistry 2, what to be our main objective is to evaluate and identify the different, shall we say, evaluation tests, okay, quantitatively and qualitatively of a certain natural products. How do we categorize, classify our certain drug products or crude drugs based on our evaluation? Now, looking at all that we have learned from our chemistry, in lieu with our, shall we say, phytochemistry, are quite similar. They may have quantitative, it may also have qualitative. And what is the difference between the two? Is that quantitative actually measure. Okay, there's a value. Okay, that certain percentage, ratio, value. There's a computation. Okay? But what to say qualitative, it identifies in terms of the color, the texture, no? The quality of the product. No? It depends upon, for example, if the product is rancid, the color deterred because of oxidation, no? And other factors. Okay? So, why do we need to evaluate and identify the different natural products? Of course, it's very obvious. It actually seeks the presence of the purity of a certain product free from adulterants. Okay? Free from adulterants. And whether or not a product is pure or impure, depending upon our evaluation of a certain product. Okay, now, what are those evaluation? It's either microscopic or macroscopic point of evaluation. Now, if you look at this, it's all about the safety, quality, and effectiveness of our product. Now, if our product is not pure, then there is a question of the safety, efficacy, and effectiveness of a certain natural product, right? And looking at if the product is impure. Now, we look at, like example, we have some impurities coming from insects or impurities due to adulterants or admixture of substitution of a low-grade product or contamination of a heavy metals. Now, we are questioning in terms of the quality of the product and, of course, the purity of the product. And this purity of the product are based on what? Standard. And that standard are coming from natural products, which are your national formularies, monographs, pharmacopeias. So you just refer it based on what is the standard. Okay, what is the standard? And how can the product be degraded or adulterated? It's either number one, there are defective collection. So what do you mean by defective collection? That is, wants to collect a certain natural product or a certain plants coming from a natural source. Is the question mark is that, is the product actually located in an area wherein it is what? In the natural source, free from any contaminants, organophosphates, heavy metals, or even pollution from any factors. Okay, like example, in the industry, or in the road, or urbanization. What do you mean by urbanization? Meaning that is, no, you try to plant in from a condominium or from an apartment wherein a lot of people. No? So this may be what? Not free from its natural source, from a natural environment, from its natural soil. Okay? And of course, intentional adulteration, meaning you substitute a product, you add a certain product to save in terms of the cost. Now we look at another factor, deficiency preparation of the drug. Meaning, like example, in terms of roasting of a caffeine seed. No? So, for example, in terms of our coffee seed, okay, too much roasting of it would remove in terms of our active ingredient. That is part of deterioration and degradation. Now, deficit of preparation of the drug may be due to other factors, like, for example, heat, light, especially with vitamins, and some other products, when exposed with the light, it may cause oxidation reduction, right? And, of course, inaccurate storage of the crude drugs. So, if you try to look at storage, are very important in terms of what? In terms of the lifespan, in terms of stability of your natural products. Okay? That's the main responsibility of the pharmacognosist or the scientist. Now, which one do we refer to? The monographs. Like example, ash value, like example, extraction values, like example, morphological characteristic via microscopic evaluation, and other evaluation as mentioned later. So there are 5 basic methods in using it to identify if the product is pure or not, and is it within the standard. Number 1 is using our senses. That is known as organoleptic evaluation. Okay, using senses, meaning that you can taste it, you can feel it, the texture, you can actually see the color, and evaluate it based on some other factors that you can see, you can hear, and you can feel it using the taste or your skin. And we have microscopical evaluation, using microscope, of course, to identify the purity of the compound microscopically. If it is added up with another admixture or substituted with another product. Another one is the physical evaluation. Okay? Whether it is, for example, the edge, the apex, and other physical characteristics. Now, we always look at chemical evaluation. What is that chemical evaluation? Like, for example, our phytochemical screening, one of our qualitative analysis, and also using TLC, thin layer chromatography, HPLC, for example, NMR, mass spectrophotometry, etc. And we have biological evaluation that will be done in our experiment number 5. Actually, in our experiments, we have done all of this one. Okay? We have done. So, because that's the main role of the pharmacognosist, if you want to evaluate a certain natural products from the head until the toe. Okay? And number 1, to digest what we have learned, organoleptic evaluation is identification in terms of what? Using senses to evaluate color, shape, size, taste, and special features. Okay? Have you done this one in your lab? Or have you done this one when you go outside and feel and see the color? If it is dry, if it is not dry. For example, feeling in terms of the texture, the color, the shape of it. To identify what? What's the main purpose of organoleptic evaluation? What? Letter A. That is to identify in terms of authenticity of your certain product. Okay? If it is real or not, based on the sample and based on the standard. So you try to evaluate it, compare if the standard is similar with your what? With your sample. Okay? So do not worry about the standard because all of it is written in Pharmacopeia. All of it is written in the monograph. And of course, like for example, Japanese monograph, for example, other monographs throughout the world, they have their own set of what? Herbal products, natural products which are listed based on their book. Okay? Monograph. And what else? This is the most simplest and very common before you move on to the chemical evaluation. If you have seen, if you have smelled, there's no aroma. And of course, it is based on organoleptic evaluation. Why you want to proceed through chemical evaluation? Where in fact, it is already failure in terms of what? In terms of your organoleptic evaluation. It will not be a waste of your time if you have evaluated first based on senses before you move on to the chemical evaluation. What do you mean by senses? Is it the feeling only of 5 senses, or your intuition is part of your senses? So that guy is really, very good, and you try to test him. Okay, some questions and all, right? That is testing. If you want to have your boyfriend or girlfriend, you always test the person. You come late, I'm sorry, I'm late. But you already been here for 1 hour already, waiting for your guy, your perfect, ideal guy, something like that, right? So you come late and I'm sorry. What will be his reaction to it? It's okay, it's okay. Now the next thing, it's okay, it's okay. The third thing, consistent. No? But if it is consistent, it flare up and all, I really hate you, something like that, then it's not consistent anymore, right? So it's like testing someone. That is what we call organoleptic evaluation, right? So that is the description, whether the characteristic is there in terms of odor, in terms of taste, that can only be described by senses, okay? So that is found to be very significant in pharmacognosy. And in terms of the size, we may say in millimeter in terms of the scale. And in the shape, you can see a simple compound, shape of the lamina, flower, fruit, whether seed. There are different descriptions. We look at the different descriptions of our plant, whether the apex, whether the lamina here or the veins of it, the vein is actually proportional to what we have characterized. Okay, so it should have its main characteristic. Okay, so we have elliptic, ovate. I hope you try to memorize this one. At least a little bit familiarization of what are the characteristics. So, simple and compound leaves, we have the main difference, odd and even, in terms of the shape of it, okay? And the palmates, like marijuana type, but the marijuana has what? The kind of a shape like this one, okay? And, of course, another one, the shape of the fruits and the seeds. We look at samara, the maple, akon of the oak, samara, elm, and of course the cone here. Okay, where is the pine? The pine tree. Where is it categorized? Okay, cone, pine. Here, and we see the wing seed. Okay, every individual. And we have hairy seed, the willow. Okay, we have the drupe, the cherry. Okay, and hackberry. These are very foreign. I haven't seen this one in the actual. Okay, so the shape of the seeds, we have, these are main characteristic of a bean seed, right? Here, okay, John and the beanstalk. Okay, narrow kidney shape. We have broad kidney. We have ovate. We have narrow ovate. Okay, so those which are studying about the seed, you can characterize based on the shape. Even how small it is, you can put it in a microscope to identify these particular shapes. Okay? Next would be sensory evaluation, whether the color, okay, of course, the color changes as it is oxidized. Like, for example, vitamins, right? When you put it in the lime, what did happen? The next day, for a couple of hours, it will be turned from orange to yellow or light yellow. Okay? We have odor. It becomes rancid, so like a rotten egg, if it is quite some time, especially with natural products. In terms of the taste, like, for example, bitter gourd, okay? and bitter taste, sweet taste, like honey, sour, lemon. That's why you call your sweetheart as honey, because sweet. But if it is a long time already and you're quarreling, it's already bitter. No? So it's not anymore honey. It's already what? Bitter gourd. Okay? So that one. Next will be texture and fracture. Of course, we say touch is quite smooth. Okay? Fracture, later on we will have a challenge. Okay? To taste it, maybe I'll put something, maybe a stone perhaps. Okay, how do you smell it? In the microscopic evaluation, the difference between organoleptic is what? Microscopy. Of course, you use microscope to identify, or magnifying lens to identify in terms of what? The identity, the purity of your certain compound, okay, or natural product. Okay, so we have here. So we have transection, longitudinal section, which are very important in a microscopic evaluation. Is this a quantitative or qualitative? Qualitative, because we are not measuring anything. Unless otherwise we are counting for what? The number of what? Stomata. Okay, the number of what? Lycopodium. We have also the number of, like example, guard cells, or vein, etc. Vein-islet, depends. We have also, this one is a quantitative microscopy. So identifying in terms of the stomata, the guard cells, and of course, vein-termination. These are very technical terminologies. But what is it? These are actually part of the structure of your plant. Okay? Don't be confused with it. Stomata, vein, palisade, these are located in terms of the lower portion of the mesophyll. So these are Lycopodium spore. So Lycopodium spore are actually been developed in 1933 by Wallace and Dervar. But this one is this particular plant, Lycopodium, Lycopodiusae, these are with specific, this one, Lycopodium. Okay? So this is actually one of the plant to identify. So what is the main purpose of this? The main purpose is what? Identification. Identification of a certain plant, because every plant has its own characteristics. Okay? Characteristics. So you can measure like Lycopodium spore method only for a certain plant which are under with this family. Okay? Next. Any question with this? So you will see that these are parts of the plant microscopically. Okay? So counting the number. Okay, we have physical evaluation. In the physical evaluation, this is more into your thesis. Okay, what is in the physical evaluation? So you would say, what is in your thesis? You would say, what is the physical and chemical characteristic of, like example, Solanum lycopersicum. That one, tomato. Okay, or so to say, Cassia lanata. That one. Then you will have. So always the students in the research, they have difficulty. They're always repeating the scientific name. They're always repeating all of it, even the generic and also the particular scientific name. So which are sounds, if you want to use the common name, would be good. In the very beginning, you can actually use the scientific name with the parentheses of the common name. Okay? Or vice versa, depending upon the format of your of the university, whether or not usually the parenthesis is the scientific name. Now, if that to be in terms of the research. So most of the students does not know how to identify the research question. What is it in your research question? And one of the research question is, what is the physical and chemical characteristic of a certain plant? Okay? Because it should reflect on the methodology. Okay? So if you have a question, what is a physical and chemical characteristic, one of your tests should be this one. Okay? Physical evaluation. And what is a chemical evaluation? Meaning it is also identification of what is it in here. This is the chemical evaluation. Example is your phytochemical screening, and of course identification, or you isolate the percentage of your certain plant constituents, no? Whether it's flavonoid, alkaloid, no, or saponins. So these are some of the evaluation tool. No? And looking at the physical evaluation, number one, these are the physical evaluation. Number one is determination of the foreign matter. Why do you need to identify in terms of the foreign matter? That is what? Number one, again, purity. Number two, identification of the characteristic. Whether or not you are identifying the correct, okay, the correct plant. Okay? If you don't have a correct plant, why you do biological? Why you do chemical characteristics? Why you want to waste your time in terms of moving forward if it is not the correct plant? Now, in terms of your research, what would be your number one in terms of evaluation of the plant? Your number one is what? Are you going to collect the plant easily? No. Number one should be what? Authentication of your plant. No? Why is it we need to authenticate the plant? Where do you want to authenticate? In UPM, okay, UPM, or shall we say Botanical Center in Malaysia, no? And soon to be MSU will be going to have authentication process here, no, in MSU, no? So in that sense, like for example, FREEM will also doing authentication. Okay, you need to authenticate. Why? Now, before you move on to your oral presentation, or your, because the panel will say, is that the real plant? Or during your final defense, are you sure that the plant is really mangosteen or not? Okay, maybe it's a calamansi, or maybe it's a other one, no? It's a grapes, or maybe it's another plant, right? So even though you know that that is a plant, but in the journals and in the manuscript, they need to have a proper authentication number to identify your plant. Okay? That is how first we need to evaluate. Okay? So authentication would be the number one process to evaluate the authenticity or the identification of your certain natural products. Okay? So that is all about determination of the foreign matter. So when I say foreign matter, what particular physical evaluation we use? That is very easy, would be the microscope. Okay? Microscopic evaluation would be the number one. Why? Because it's easily can be identified, no? If there are foreign matters which we cannot see with our senses or in our naked eye. Okay? It may be through organoleptic evaluation, but certain foreign matters, when it is mixed together, may not be, no, visible in our naked eye. Okay, that's the problem, right? So we need to use microscopic evaluation. Now, another one is determination of the ash or the ash values. No? There are different identification of the ash value, but what is the main purpose? The main purpose, again, going back to what we have learned, is the purity and, of course, the identification of a certain pure compound, okay, or your natural product. Okay? Looking at also some which are not. Well, how what's the process of ash value? Okay, what is this process? You put it in the what? Crucible, and you try to heat it. It's like what? Okay, it's like a dead body that you need to what? Put it in the ash, right? What is that name? Cremate. Okay, you cremate it. But of course, we don't put the name cremate in that ash value. Of course, we need to put it into ash, application of heat in the crucible, make it into the ash, and we count the what? The ash, and of course the sample before it is put into an ash. Okay? So that is what we call as the total ash value. Okay? So put it in the crucible, okay, in a certain container which does not affect with the heat. So like this one. Have you seen the crucible? No, crucible, that one. And you put it there, and what will happen is it becomes ash, and you count the number of in grams with an ash versus the one that you put, the powder or your particular powder, dried powder. Okay? Then after that one, let me example, sir, there are certain inorganic compounds that cannot be what? Cannot be decomposed or degraded. Like for example, your certain products that are inorganic, sodium carbonates, silica, calcium carbonate, which are difficult in terms of, shall we say, making it into an ash. So what will happen is you put some solvents. And that solvent may be your diluted hydrochloric acid. Okay? So that one. That is what we call as acid insoluble ash value. Now, we try to replace it with sulfate. Okay? So uric acid, then that's called sulfated ash value. Okay? If that will be water soluble ash value, like for example, combination of chloroform and water, then that will be water-soluble ash value. Okay? Very easy to remember. To actually identify your ash values. Okay? That one. Any question with this? So it will identify based on monograph or pharmacopeia the standard ash values. Then based on your computation, then of course you try to relate it, correlate it. And what will happen is if it is up and down, there are values below or up with it, then you will identify impurities of your certain product. Okay? Next, we have mucilage here to identify the mucilage by swelling factor. What is that swelling factor? To identify in terms of the purity of your crude drugs, and this is a test for those content of mucilage or mucilage. Okay? So, like example, thick gelatinous substance which are composed of this. So, identifying the mucilage content. Next, we move along. Okay, we move on to extractive values. What do you mean by extractive values? That is the one, is either there are two processes for extractive values. The application of hot and cold. What you actually do in the lab is a what? Hot or cold? Hot or cold? Maceration. Cold maceration, right? So application of alcohol, whether it is 90%, 95%, 60, 50, some of you absolute alcohol, or maybe vodka. All into one. So, in that sense, you are adopting this kind of extractive value. There are two processes. The one that you do in the lab is a cold maceration, wherein you add up alcohol or water or whatever, and solvent. And another one is hot maceration, which you need to have using what? Using reflux. Okay? Using reflux, adding up heat. That is what we call as hot maceration for about 6 to 12 hours in time. Okay? So, this is to extract certain component. And these are divided into water-soluble extractive values. Okay? We have alcoholic. These are water. Very obvious. Alcoholic. Alcohol. Okay? We have ether. Okay, so that one is either chloroform water. Okay, can be. Okay, this one is more onto alcoholic. Okay? Whether it's ethanol, methanol, this is here. Okay? So what you're going to do is that you solve what is then the grams, okay, of your extract, okay, the residue of it. Okay? So in the computation, you will say you try to have the percentage of your certain extract, the one that you weigh, okay, the one that you weigh in terms of the powder or not powder of your extract. When you add up your alcohol or water, then for how many days, the value is still there. And of course, once you put it into the water bath or in the water evaporation, then what is the gram? Okay, the gram of your extract without alcohol already. Okay, the gram of your residue, the gram that you have extracted from the plant. Okay, that is the meaning of the extractive values. Okay, usually us, we need to compute this one to identify in terms of the concentration of your certain product, natural product. Okay? Especially if you undergo like little dose 50, for example, with effective dose, toxicity testing, for example, biological testing, you need to have this one. Okay? And this is a study according to Chandel et al., according to Pharmacognosy research about standardization of some herbal anti-diabetic drugs in a polyherbal formulation. You can see here the different solvents: water-soluble, alcohol-soluble, ether-soluble. And most of the product have, like E. officinalis, have higher water-soluble extract compared with ether-soluble extract. And what is the meaning of this graph? It actually says that most of the products in the market are affected in terms of different factors. One of the factors is the process of extraction, and another one is what? The application of heat, and of course the methodology, the process. Okay? There are some, like for example, one study which is papaya is good as an antimicrobial. There is a zone of inhibition more than 10, intermediate. But one of the study which is happening now is actually there's no effect. There's no effect of the papaya. So what will happen? What do you understand of it? It means that there is a problem in terms of the extraction process. And this is what I actually experienced when I was doing my master's and even in the PhD, or even other research with my RP2 students, that, sir, there is no zone of inhibition. What should I do? How about your extract? Can I see your extract? What will happen? Very, very diluted. How can you have a certain zone of inhibition wherein the extract is very diluted? I said you better rotary evaporate it, or you put it in a water bath so that it will be more concentrated. Okay? The concentration is very important. That is why you are complaining with your supervisor that there is low efficacy and lower therapeutic efficacy with your rat or within your mice. Right? Because of this. Because of the process or methodology of extraction and the use of the improper solvents. Okay? Not good solvents. Okay? So it's very important to identify also the very good solvents that we have. And we have physical evaluation, okay, using analytical method to identify the quantitative analysis. Okay, like thin layer chromatography. We have also UV vis, and etc. These are under with analytical methods. Okay? Next will be chemical evaluation, mainly on phytochemical screening. Okay, identifying whether or not our product is test for carbohydrate, flavonoids, and etc. What's the main purpose of phytochemical? Is to identify the primary and secondary metabolites, and to distinguish our product is pure, and identification of the certain constituents or phytochemical constituents of a certain product or natural product. Okay? So that's why we always do phytochemical screening. Sir, all of the literature, they said they've done phytochemical screening. Then do not do phytochemical screening. You can do chemical tests, another chemical test, or physical evaluation using thin layer chromatography, or what? HPLC or higher level. Okay? Higher level of screenings. Okay? So what's the important of the screening? Thin layer chromatography. What's the main purpose? For example, there was a study about the henna. Okay, henna, from the henna leaves, am I correct? So, in that sense, if this product, the natural product of henna, is adulterated with the normal ink, because very costly, the henna, right? Am I correct? So, in that sense, they identify using thin layer chromatography. What will happen? You can easily see it in a thin layer chromatography. And of course, the purity of the product. Okay? Because the ink would have RF value, no, same as with the sample. So that's the problem. So if our product is substitution or admixture, no, if the product is adulterated. Okay? So you can have research on that, specifically on different angles. No, like example, this would be leading to what? Number one, public health. Okay, because if it is not pure, what would happen? Then safety, efficacy of the product would be compromised. Okay, public health is very important. Now, looking at pharmacists, how can we contribute to the society? Especially, like for example, me, I am studying on heavy metals, okay, of natural product, unregistered product in Malaysia. No? Aside from that, you can also like study on the food. Like for example, nowadays there's a news that the shrimp is contaminated with chloramphenicol and nitrofuside, something, a certain drug antibiotics, okay, which are in the market. Okay, so these are in terms of public health issues. Next would be in terms of chemical evaluation. Please do familiarize yourself with the phytochemical screening, especially with secondary metabolites. Okay, these are very important and these are very useful for your examination. Yeah? So, carbohydrate, like primary metabolites, Molish test, Benedict test, Felix, alkaloid, Wagner, Dagendorf, no? It's very UK thingy, that one, and etc. No? Shinoda test, so Japanese, no? Flavonoids, etc. So these are very important for evaluating, and we will do this one on Thursday and Tuesday for phytochemistry too. Okay? Next would be biological evaluation. How do we identify evaluating it biologically? Is either using models. What are those models? Like, for example, for antimicrobial, you use with what? Bacteria, fungi, or shall we say helminths, worms, and other models, subjects. And we have also like anti-diabetic. Before I'm doing anti-diabetic of a certain product, like for example also hypertension. My students allowed to do like for hypertension, diabetes, cholesterolemia. I have studies on that also using animal models. Now, when they approach me with the rat and all, I say, I will do it. I will do it. Okay, because it's very clever. Sometimes when you hold it, it will bite you and all, no? But you need to be friendly with them. No? There's a training with that in terms of animal handling. If you want to go, this is very interesting, I will tell you, no? Because most of my students, I am also doing animal studies, antipyretic activity of anthropogon, no? When I was in bachelor with this, and we inject, no? We try to feed with this natural product, and we see that there's a decreasing level of pyresis, no? With these rats. And it's not a normal rat. It's a big rat, no? That one. Thanks God it's not black, no? So, that one. Okay? So, it's very important. If you do in your bachelor, some of the animal models, that will be very good. One of my students in FHLS, she's doing wound healing. Okay? Wound healing in terms of the animal models. Okay, that's very interesting. I said, your product's very good. Now test with this one for wound healing. That's very good. And she has a result, a very good result. No? That maybe, counteract with another product, which is very common in the market. This is recorded, so I cannot tell you. So, it will be very good. No? Because wound healing is still very controversial research nowadays. Right? Okay. And these are some of the biological evaluation. Anti-diabetic activity, we have analgesic. Okay, these are using animal models. Anti-cancer activity would be, like, for example, cell lines. Like, for example, one of my students in a master, she's doing psoriasis. Okay, and she has cell lines of psoriasis coming from America. Okay, it's in the US, and she's doing it fine, very good. No? One of my friend who is a PhD, she's doing with cancer in the skeletal muscles, and it was terrific, very good. No? Injecting this cancer lines with animal models. So the rabbit actually had this kind of disease, and he tried to extract it. Using with this one, and it has a very good research. And this anti-cancer have a high impact in journals and publications. Okay, anti-ulcer activity can also be done. These are very excellent research because ulcer is very common and very, it's very painful. Now, whether ulcer in the what do you call it, esophageal line, or is either in the oral cavity, or is either in the stomach. Okay, it's depends upon it. Okay, so that is some of the evaluation that we need to take to remember as we go along with our studies. And I think we will have 10 minutes break.